Hello everyone, my name is Fan Bao. My research proposal title is Regulation of Circulating microRNA Following Acute Aerobic Exercise in Obesity. As is known to all, obesity is a big problem nowadays. Recent studies have shown that the worldwide population for obesity has reached 2.1 billion, and more than 35% of US population were obese in 2010. Usually, obesity is determined by a person's BMI or body mass index. It is calculated by a person's height divided by his weight squared, and all of the units are in metric system as meter and a kilogram. Based on the BMI table, a person is considered obese if his BMI is above 30 and normal if BMI is below 25. One of the major reasons that obesity is seen as a serious problem is that it can lead to various diseases. Generally speaking, the initial stage of this path starts with an increased BMI, waist circumference, and waist to hip ratio. Then it progresses into high blood pressure, high blood glucose level, dyslipidemia, insulin resistance, inflammation, and etc. Eventually, the worsening of these physiological signs give rise to actual cardiovascular disease. It has been reported that 28 million deaths are caused each year by obesity and its related diseases. MicroRNA or miRNA is a useful biomarker related to obesity research, and it serves a major role in the research proposal. miRNA is a type of single-strand RNA molecules, usually about only 22 nucleotides long. Since it is an RNA molecule, it is produced in the nucleus via the transcriptionary machinery from its specific miRNA gene. After transcription, the precursors of miRNA undergo a series of modification processes within the nucleus, and eventually the mature miRNA is released into cytoplasm, where it exerts its power on mediating messenger RNA translation and protein production. miRNA is involved in numerous biological processes, such as those related to obesity and inflammation. One type of miRNA is called circulating mRNA or ci-mRNA. Unlike regular mRNA that resides in cytoplasm and is easily degraded, ci-mRNA exists in bodily fluids such as plasma and is stable and easy to detect. This makes ci-mRNA a very good target for extraction and quantification. Some ci-mRNAs are specifically associated with inflammation. For instance, MIR21, 130B, 221, and 222 have been shown to be pro-inflammatory, whereas MIR126 anti-inflammatory. It has been well established that exercise, especially aerobic exercise, is very beneficial for obese individuals because it can confer anti-inflammatory effect on the body. By sticking to a proper aerobic exercise plan, study shows that the risk for cardiovascular disease and type 2 diabetes could be significantly reduced. Interestingly, recent studies have found that in normal weight individuals, ci-mRNA are also related to aerobic exercise. In particular, the plasma level of the following ci-mRNA, MIR21, 126, 130B, 221, and 222, can be regulated by acute aerobic exercise in normal weight individuals. This list of ci-mRNA is exactly the same as the list of ci-mRNA associated with inflammation. This means these 5 ci-mRNAs might serve as a link between exercise and a physiological inflammation response. In other words, regulation of these 5 ci-mRNAs might represent the anti-inflammatory effect bestowed by acute aerobic exercise. And the anti-inflammatory effect of acute aerobic exercise could potentially be measured by the change in these 5 ci-mRNA expression. However, one question now pops up. The link between ci-mRNA, exercise, and inflammation has only been reviewed in normal weight individuals. Then how about obese individuals? Therefore, two questions naturally come forward. First, do the same inflammation-associated miRNAs also respond to acute aerobic exercise in obese individuals? And second, if they do, were their response in obese individuals different from those in normal weight people? And these two questions are the basis of the research problem in this proposal. The research problems are significant because, first of all, obesity is a big problem, and any attempt to address the issue does a great service to reduce the detrimental impact obesity has on humankind. Second, although aerobic exercise has long been recognized as having treatment effect on obesity, there has been lack of information regarding how such treatment effect could be measured. Third, if aerobic exercise treatment effect could be quantified by way of biomarker, it will be possible to not only predict the treatment effect on an obese person's inflammatory profile, but also individualize the dosage of exercise treatment, such as volume and intensity of exercise, to better suit the need of each individual patient. Finally, inflammation-associated miRNA have been shown as potential candidates for biomarkers that could serve as predictors of the exercise treatment effect. However, more research is needed in this area, and that's why the research problem of this proposal is extremely important. The purpose of this study is twofold. First, it is to determine whether acute aerobic exercise will regulate plasma expression of inflammation-associated circmRNA in obese individuals. And second, it is to review whether exercise-induced cirmRNA response in obese is different from normal weight individuals. Accordingly, there are two working hypotheses corresponding to each of the study purposes. Hypothesis 1 is that inflammation-associated cirmRNA plasma expression will change in obese individual after acute aerobic exercise. And hypothesis 2 is that the exercise-induced expression of inflammation-associated cirmRNA will be different in obese compared to normal weight individuals. Finally, before we go into details of literature review and experimental methods, it is highly necessary to look at the definitions of certain terms that might appear unfamiliar. VO2 max in this proposal means maximum amount of oxygen a person can utilize during very high-intensity exercise. Acute aerobic exercise in this proposal defines a one-time aerobic bike exercise at 75% of a participant's VO2 max. Exercise-induced cirmRNA expression in this proposal means total amount of exercise-induced cirmRNA expression relative to baseline or pre-exercise level calculated with a method called area under the curve relative to increase, or AUCI. Here, AUCI sums the total area under the time concentration curve relative to the baseline level as a representation of total cirmRNA expression throughout exercise and recovery time. It can compare the relative change in cirmRNA concentration post-exercise between obese and normal weight individuals. Here in this figure, for example, the level of M1 represents the baseline. So AUCI of this time concentration curve will be this shaded area. Limitation of this research proposal is small sample size and untruthful self-report about health history. Due to budget, time, and manpower restrictions, the study will likely not be able to sample a large number of normal weight and obese individuals from various locations to come up with a conclusion generalizable to the general population in the U.S. In addition, the subject screening process requires that all subjects, obese or normal weight, have to be free of metabolic syndrome. However, since the health history will be self-reported, there is a chance that some participating subjects who claim to be healthy actually either remember wrongly or are unaware of their metabolic syndrome. In that case, the results of the study could potentially be biased due to the confounding factors of diseases. Delimitations including choosing to examine only disease-free obese individuals rather than having no inclusion criteria for recruitment. The reason for such choice is that obesity-related diseases alter a person's inflammation profile dramatically, and hence any change in inflammation-associated cirmRNA expression could be due to the diseases rather than exercise. Thus, in order to eliminate as many confounding factors as possible, the study decides to recruit only healthy obese subjects. In addition, the study also chooses to use exogenous spiking miRNA to calculate miRNA concentration, rather than internal positive control. This is because normalization based on internal positive control only gives relative fold difference of cirmRNA compared to the control. Since it is currently unclear whether the commonly used internal positive control remains stable after exercise, the normalized cirmRNA expression might not be accurate. On the contrary, using a well-controlled exogenous spike-in not only ensures a stable standard to normalize cirmRNA expression, but also allows for calculation of absolute concentration rather than fold difference. Now let's take a look at literature review. Low-grade inflammation has been recognized as the leading cause of obesity-related diseases. A few biomarkers have been identified for inflammation, such as tumor necrosis factor alpha or TNF-alpha and interleukin-6 or IL-6. Recently, inflammation-associated circulating microRNA has shown promise to be a new kind of biomarker for inflammatory response. As mentioned earlier, cirmRNA is stable in bodily fluid and easy to detect. Studies have shown that some cirmRNAs are related to obesity while others to exercise. Among the obesity and exercise-related cirmRNAs, 15 of them appear to be related to both obesity and exercise. And among the 15, 5 of them are also linked with inflammation, and they are MIR21, 126, 130B, 221, and 222. MIR21 has higher baseline level in obese than normal weight individuals. MIR21 is characterized as pro-inflammation because it has been shown to bind to toll-like receptor to induce IL-6 secretion in cancer cells, and it can also be mediated by IL-6 in inflammatory processes in human keratinocyte cells. Interestingly, since MIR-21 can both induce IL-6 expression and be regulated by IL-6, there might be a positive feedback between MIR-21 and IL-6. MIR-126 has lower baseline level in obese than non-obese individuals. MIR-126 is considered as anti-inflammation because it inhibits VCAM1 in endothelial cells in order to prevent adherence of leukocytes. It can also downregulate chemokine ligand 2, or CCL2, which is a key cytokine promoting inflammation in obesity. MIR-126 can also be upregulated by exercise, and its elevated level could counter the development of diabetes. MIR-130B has lower baseline level in morbidly obese people than obese individuals, but its baseline level in obese is not different from normal weight people. MIR-130B is seen as pro-inflammation because it is upregulated by TNF-alpha via the NF-kappa B pathway. It can also suppress adipogenesis and promote adipocyte dysfunction. MIR-221 and 222 have very similar functions due to their being homologous. However, MIR-221 has a lower baseline level in obese than non-obese individuals, whereas MIR-222 has the opposite effect. Their role in inflammation is ambiguous. One study shows that both are upregulated by TNF-alpha in mouse adipocyte. Yet another study shows that the two mRNAs are downregulated by TNF-alpha in human adipocyte. Yet another study shows that the two mRNAs do not change their expression when exposed to TNF-alpha in human vascular endothelial cells. Therefore, MIR-221 and 222 might have different roles in inflammatory response in different cell types. Next, I will discuss the method section of the research proposal. For participants, the same number of obese and normal weight individuals will be recruited. The selection criteria are listed here. First, subject must be untrained because previous training could affect a person's physiological response to acute aerobic exercise and thus bias the expression of CI-mRNA. Therefore, those who report larger than 150 minutes of moderate and high physical activity per week will be excluded from participation. Second, subject must be healthy. Prior disease is another confounding factor affecting a person's physiological response to exercise. Third, subjects must be from 18 to 35 years old to limit age's effect on experiment. Fourth, women who are pregnant, nursing, or taking hormone replacement therapy will be excluded because their inflammatory response could be altered due to those scenarios. Fifth, subjects are considered obese if his or her BMI is above 30 and normal weight if BMI below 25. And finally, subjects will be excluded if they use any tobacco products or consume an average of 10 or more alcoholic beverages per week. 1 day prior to each lab visit, subjects must fast overnight for more than 8 hours and refrain from alcohol, caffeine, and intense physical activity. These efforts are for the sole purpose of reducing potential confounding factors of the experiment. The procedures of the experiment are shown in this flowchart. Each subject makes 2 lab visits. For both visits, subjects will arrive at the lab between 7 a.m. and 9 a.m. The first visit will be to inform subjects the experimental procedures and acquire their consent. Subjects will also familiarize themselves with instruments, undergo anthropometric parameter assessment, and conduct a treadmill test for VO2 max. VO2 max protocol is as follows. It will start with a 3-minute warm-up at 3 miles per hour with no grade, and then the speed will increase in order to reach 80% plus minus 5 beats of the subject's age predict maximum heart rate. Then this 80% of maximum heart rate will be maintained for 4 minutes. After that, the grade will be increased by 2% every 2 minutes, and the speed will be constant. This will be continued until voluntary exhaustion. The test should be completed within 12 to 15 minutes. Both heart rate and RPE will be recorded, and a standard criteria to determine whether true VO2 max has been achieved will be used. The second visit is 1 week later, in which subjects will complete a 30-minute continuous exercise on a treadmill at a 75% VO2 max. Whole blood will be drawn at 4 time points, prior to or at the baseline, immediately post, and recovery at 1 hour, and 2 hours after the exercise. 10 milliliter of blood sample will be collected from the antecubital vein using a 21 gauge butterfly needle into a tube containing K2-EDTA. After collection, blood sample will be centrifuged immediately at 1000 G for 20 minutes at room temperature. Plasma will be collected after centrifugation and stored at minus 80 degrees Celsius in 500 microliter aliquots for mRNA assays. Total RNA will be extracted from 200 microliter of plasma with QIAGEN miRNeasy serum plasma kit following the manufacturer's protocol. The reliability of RNA extraction processes is vouchered by QIAGEN company. Normalization of sample-to-sample variation in miRNA quantification will be achieved by spiking in C. elegans miRNA 39 or cell MIR 39 to each plasma sample during extraction. The absolute concentration of plasma miRNA will be normalized by the extraction coefficient calculated from the starting and recovery spike in cell MIR 39 concentrations. The validity and reliability of this normalization procedure are corroborated by previous literature. A 5 microliter of extracted miRNA will then be reverse transcribed with a TaqMan microRNA reverse transcription kit and each respective primer according to the manufacturer's protocol. Additional cell MIR39 miRNA will be reverse transcribed to serve as standard curve for real-time PCR. The reliability of reverse transcription process is supported by ABI company. Subsequently, RT-PCR will be used to quantify each target, spike-in, and a standard curve miRNA with TaqMan microRNA assays and their corresponding primer probes following manufacturer's protocol. Target samples will be analyzed in triplets, spike-in, and standard curve duplicates. The validity and reliability of RT-PCR procedures are upheld also by ABI company. Data analysis will be performed with the Statistical Package for the Social Sciences, or SPSS, version 20. Data to be analyzed will be the absolute concentration of each CI-mRNA. Depending on whether the values of absolute concentrations of miRNA will conform to normal distribution, log transformation might be warranted to allow for proper statistical analyses. The data will be formed into 2 groups, obese and normal weight, and 4 time points, pre, post, recovery 1 hour, and recovery 2 hours. To assess the first hypothesis, a one-way repeated measure analysis of variance, or ANOVA, with 4 time points and with 4 time points within obese subjects will be utilized to examine whether there is a time effect on plasma levels of MIR 21, 126, 130B, 221, 222. If, for a specific CI miRNA, ANOVA reports a significant difference among 4 time points, or the p-value is smaller than 0.05, then it means exercise will induce change in plasma CI miRNA level in obese individuals, and this will support the working hypothesis. And after that, a post hoc comparison will also be conducted to examine which time point elicit the highest and lowest CI mRNA expression. However, if p-value is larger than 0.05, then there is no time effect and exercise does not induce change in plasma CI mRNA level in obese individuals, and this will lead to the rejection of working hypothesis. To assess the second hypothesis, independent t-tests will be used to compare the differences in AUCI for target CI mRNAs between obese and normal weight individuals. For a specific CI mRNA, if independent t-test reports a p-value smaller than 0.05, then there is a difference and the amount of change in plasma CI mRNA level induced by exercise is different between obese and normal weight individuals, or otherwise the working hypothesis number 2 is supported. However, if the p-value is larger than 0.05, then there is no difference and the amount of change in plasma CI mRNA level induced by exercise is not different between obese and normal weight individuals. Or in other words, the working hypothesis 2 is rejected. And that's the end of my presentation of the research proposal, and here are the references. Thank you for watching.